cells use global regulatory systems to monitor and regulate what's happening with regard to nutrients that they need. The main nutrients that cells are looking for include carbon, nitrogen, and phosphorus. We've already talked about carbon, mentioning the catabolite repression system, which allows a cell to determine whether or not glucose is present and repress the use of other carbon sources until the glucose is exhausted. And now we're going to turn our attention to nitrogen, because nitrogen is the other main component that cells need to be acquiring, and they're going to look for ammonia first. It's the preferred nitrogen source. And they have other plans that they need to invoke in case ammonia is limiting. So we're going to talk about a couple of components. RPON is the gene that encodes the sigma 54. This is a special sigma factor for nitrogen systems, and we talked about it earlier in the semester, and we'll talk about it again briefly here. We'll also talk about NTRB and NTRC. These are the genes that encode a two-component system, regulatory system, that will be involved in this process. And finally, they're going to, along with a couple of other proteins, be involved in regulating the expression of glin-A. And we'll look at that in just a second. This cartoon shows how the NTR system works for nitrogen assimilation. So assimilation means, just break it down into two words, as similar. So it's the way that the cell will take whatever nitrogen, carbon, and incorporate it into cellular material, so it becomes part of the organism then. Well, the key components to think about here are glutamine. That's really what the cell is monitoring, whether or not glutamine levels are high, which is shown on this side, or low, because we're looking at this bar. So let's look first at the situation where the levels of glutamine are high. If glutamine is high, we're looking at this part of the arrow here, and we're going to convert a complex called P2 in the cell to a form where it's not modified. So let me back up for a second and say that P2 is responsive to the glutamine levels, and there are two forms of it. This is the unmodified, and this is the modified. So again, this is a post-translational modification. We've seen a lot of these. It's either going to have UMP, and the enzyme that carries out that reaction is uridyl transferase, and that's the modified form. Or we have the unmodified form. Unmodified when glutamine levels are high, modified P2 with this UMP on it when glutamine is low. So now that we've established that, let's look at what P2 does. If it's in the unmodified state, that's the way that the cell knows that glutamine levels are high, and P2 will bind to this trimer, protein trimer of AMTB, which is a transporter in the membrane, and it prevents the transport of ammonia. It does another thing though too. It acts on AMTB, and it also binds to this other protein called NTRB. And NTRB is part of a two-component system. When NTRB is bound to P2, it actually acts as a phosphatase, and it removes phosphate from NTRC. All right, now let's look at what happens if all the glutamine has been used up in the cell, and glutamine is the favorite way of kind of moving these ammonia groups around to other amino acids, etc. So, this is an important amino acid in the cell. If the levels of glutamine are low, now that P2 is going to be modified by the uridyl transferase. So now we have the modified form, and having these UMPs on P2 precludes it from binding to the AMT-B, this transporter. So that means now ammonia will be able to come into the cell. And P2 also is not going to be able to modify NTRB. So now NTRB is a kinase, and it will add a phosphate to NTRC, and NTRC can then bind to DNA. So it's a little complicated, but again, it's a, this is a really important resource for the cell, and there have to be ways so that a cell can know when do we need to bring ammonia in. You don't want free ammonia just coming into the cell all the time. That's dangerous. So it has to be very carefully regulated. It's a beautiful strategy. Well, let's look at a situation now where NTRC has been activated. That means that the levels of glutamine in the cell are low and that the system has been activated to transport ammonia into the cell. And now NTRC is acting at the other end of this pathway to regulate expression of proteins that are going to help utilize that ammonia that's being brought in. So here's NTRC, it's activated, and it's shown over here. It's called an enhancer binding protein in this image because it actually shares some similarities with eukaryotic enhancer binding proteins, and they all have in common the fact that they bind to an upstream activating sequence, a particular DNA sequence recognized by the protein. The other part of NTRC is that it interacts with RNA polymerase when it's bound to sigma 54. And sigma 54, you'll remember, was different than the other sigma factors because it has a different spacing in its recognition of the sequences in the DNA. In the promoter, it recognizes a spacing for minus 12 and minus 24, and usually we've been looking at minus 10 and minus 35 for promoter recognition. So let's return to this. So when NTRC binds, and we've got RNA polymerase bound to the sigma 54, the NTRC actually bends the DNA around so that it can interact with a part of RNA polymerase that looks like it's at the top of the protein. It's assisted by a protein called integration host factor. An integration host factor actually participates in many similar reactions in the cell to help bend the DNA. But what the interaction with NTRC and polymerase does is it will allow then utilization of energy in the form of ATP or GTP. So that's what's shown here. We're removing phosphate to release energy, and that allows this kind of upward movement with NTRC so that we see an open complex form in the DNA, and then transcription can proceed in this region. Now let's look quickly at what NTRC looks like in terms of the protein. Here's the N-terminal part of NTRC. There's a regulatory domain. Here's the important AAA domain, which is what binds to NTP and releases energy so that we can have this stimulation of the open complex. And then finally, here's the DNA binding domain at the C-terminal end. So here's what NTRC does. We know that there are two different promoters. Here's P1 and P2 for the GLIN-A gene. There's a small amount of transcript being made to make GLIN-A all the time, and that's from a sigma 70. Remember, that's a housekeeping sigma factor. But when the cell needs to make more glutamine, it's going to increase expression of this, and that's where NTRC comes in. So this is kind of the need-based emergency backup system. We've got phosphorylated NTRC and sigma 54 This is the sequence we saw on the previous slide, and it's going to make much more glin-A. It also will have some mRNA that reads through to increase expression of NTRB and NTRC. So expression of glin-A goes up, and what is glin-A? Well, this is a glutamine synthetase. And it is directly involved then in converting the ammonia that the cell is bringing in via the transporter that we saw on the previous slide. And it will take glutamate in the cell, and using some ATP, convert that to glutamine. And glutamine then enters many pathways and donates the extra amino group to all of the other intermediates from the TCA cycle, etc., so that you can have a way of getting all that ammonia into the cell material. And again, having glutamine synthetase here, GS, allows the cell to capture all that ammonia so it doesn't build up in the cell. Global regulation processes also help to determine starvation responses in cells. We've already seen earlier a little bit about what happens in stationary phase because we have regulation by RPOS, the sigma factor. And I'm going to spend a few minutes now talking about the stringent response. And this involves genes RELA and SPO-T and a novel cofactor in the cell called guanosine tetraphosphate, or it was originally called magic spot. And the stringent response is very important for a lot of organisms because if they're out in the environment in particular, they run out of food a lot, and they need to enter pathways that allow them to persist in the environment if nutrients are going to be limiting for a long time. Sporulation responds to this. This is a bacillus cell with spores inside, the dark red spots. Myxococcus xanthus does this, and it responds, its initial response is to amino acid starvation, and the stringent response helps to activate a development pathway in which the cells form these fairly elaborate looking fruiting bodies, these multicellular structures that contain spores inside. Well, here's a cartoon that describes a good portion of what happens in the stringent response. We're going to be looking at how this response coordinates what's happening at the ribosome level with what RNA polymerase is doing. So we're going to focus on amino acid starvation. And here is our ribosome in yellow, and here is the mRNA, and here's our polypeptide being produced. Now we know that if there's a shortage of certain amino acids, that's going to mean that there's not a charged tRNA, and we end up with nothing in this chamber in the ribosome, and the ribosome is going to stall. When that occurs, we're going to activate a protein called RELA. Now RELA is an enzyme that doesn't typically participate in what the ribosome is doing, but it is there monitoring what's happening at the ribosome. And it does so because it's bound to one of the proteins in the large subunit called L11. When stalling occurs, that activates RELA's enzymatic activity. And what RELA does is it takes ATP and GTP and makes a second messenger in the cell called guanosine pentaphosphate. We'll also see that we get the pentaphosphate and the tetraphosphate form. At any rate, this is the molecule that we get. It was initially called magic spot because the people that were studying what happened during starvation noticed that when they did thin layer chromatography, they would take samples that were cells that were normally growing and spot extract from that, and they would take cells that had been starving, and then they would develop this using standard chromatographic procedures. These were, by the way, radioactive samples, and they would see a couple of spots up here in the normal sample, and if I switch to a different color, they saw a different sample that occurred on the that appeared in the cells that had been starved. So they called it magic spot, and of course they figured out structurally what it was. And then it took some time to figure out what magic spot was actually doing. But years later, work showed that it was actually interacting with RNA polymerase to regulate what RNA polymerase could do. So what would happen is that during this amino acid starvation, this RNAP bound to magic spot would shut down DNA replication, cell wall materials, fatty acids, lipids, stop making ribosomal proteins and elongation factors, and instead ramp up things like RPOS, which can control glycolysis and survival genes, oxidative stress genes, and osmotic stress genes. Other stress components were also upregulated, and amino acid biosynthesis and proteolysis were both upregulated, which makes sense. I'm not going to talk about SPO-T. It is another enzyme that can carry out the reaction to produce the guanosine pentaphosphate or tetraphosphate, and it is activated in response to stress and starvation also. On the left is an image that was from your textbook, again showing essentially the same thing, that relay is activated in response to a stalling on the ribosome. And then this is an image showing the guanosine tetraphosphate from relay that binds to RNA polymerase and modifies its activity. So again, this is similar to the other examples of post-translational modification that we've talked about. Cells also need iron. It's essential for almost all organisms except for lactobacilli, and it's unusual because iron, the available iron in the environment, is good if cells are growing anaerobically because most of the iron is ferrous at that point, and cells can bind to the ferrous form using the fur protein, which activates fur. But if a cell is growing aerobically, then most of that ferrous iron is oxidized to ferric iron, and that is going to form insoluble hydroxides that are unavailable to the cell. So that's a real problem. So let's look at a situation where the cell is growing anaerobically first, and many cells do grow anaerobically, or at least under microaerophilic conditions. So here's fur bound to iron, and it's active. It represses Rib, and other operons then can transcribe and translate proteins that will bind to iron. Now let's look at a situation where there is no iron, or no available iron, because it's all in the oxidized form. And fur is now in an inactive form, so that means that Rib will be produced, and that's shown down here. Here's Rib, an RNA with secondary structure, and it will interact with HFQ, the protein that we've already seen. And Rib is very unusual because it can base pair with certain mRNAs in the cell, but it's a very specific set of mRNAs, and they are listed up here. And what's unique about these is that these are all RNAs that encode proteins that bind to iron, but this subset of RNA operons, RNAs that are made from these operons, are for genes that are, or proteins that are considered non-essential. So essential iron proteins might include a cytochrome because you need to be able to make energy. So you don't want to shut those off, but one of the 18 targets here is superoxide dismutase. And even though it's very good to have superoxide dismutase, if you are in a situation where your iron is limiting, superoxide dismutase is dispensable. So it's going to be one of these whose RNA gets degraded. And it gets degraded because it can base pair. It has a sequence in it that will base pair with Rib. HFQ will bind. This is recognized by something called a degradosome, and that RNA will be degraded. So this is the way for the cell to know if iron is limiting. We have to get rid of the mRNA for all of those operons that encode proteins that we don't absolutely need for growth, and preserve all the iron that is present so that we have enough to make the active sites for cytochromes and other essential proteins. So I just want to recap. This is a figure from your book. And again, if we have high iron concentrations, the Fer protein will bind, and it's activated, and it will bind to the Fer operator, and it will repress transcription of iron assimilation genes. For example, the siderophores, which are the proteins that will bind to ferric iron, the insoluble form in the aerobic environments. When there's low iron concentrations, there's going to be no activation of Fer, so the Fer operator is not repressed, and we see that we get transcription of those regions, so we'll make siderophores. So again, the Fer example is an example of negative regulation, and positive regulation is going to involve the system that I was just talking about with Rib. So again, if we have high iron concentrations, no Rib is made. But if we have low iron concentrations, Rib is made, and here is the Rib. Antisense RNA, HFQ binds and causes, stimulates this binding to the RNAs in other operons that are encoding proteins that need RNA, or bind iron. And here's the matchup between those, for example. So again, here's SOD-B, the superoxide dismutase, and once this binding occurs between Rib and SOD-B, so we have sense and antisense, this is when we'll see that RNase E, that complex, the degradosome, will degrade the RNA. And here is that special region in the Rib RNA, so even when it forms this secondary structure, that region is available for base pairing with the RNA for SOD-B and all of the other operons that involve non-essential functions of proteins requiring iron. Well, let's look at an example of an organism that actually recognizes and uses iron as a way to regulate virulence. So this is Corynebacterium diphtheria, an organism that has kind of unusual cellular morphology and is actually resident in the throat of many individuals. It's a problem. Diphtheria is still a problem in many countries, Russia and India, for example, and down in the Philippines, these areas in red. not so much of a problem in the U.S. because, of course, we vaccinate people against this disease. The real disease symptoms are actually due to the production of a toxin that leads to the formation of a pseudomembrane. And you can see that pseudomembrane here because it will actually come down and cover part of the trachea and esophagus so that you actually block these passages, and people can actually die of suffocation because of this blockage in the throat. And of course you can get toxemia if the organism is in the blood, and it can damage the heart and kidneys, nervous system, for example. Someone who hasn't been vaccinated and gets a strain of Corynebacterium diphtheria that has a particular phage called the beta phage will then be carrying the tox gene. And the tox gene encodes a type of toxin called an AB toxin. The B part is the part of the toxin that binds to the host cell, and the A part is the active site, the active portion of the toxin. So B will bind and help the A portion of the toxin protein get into the cell. So once the toxin is in the cell, what it does in this particular case is it takes host NAD and it binds to elongation factor 2, and it takes the ADP ribose group from NAD and transfers it to elongation factor 2, modifying it in such a way that it can no longer function in protein synthesis. Of course, not everyone has this issue, even if you're not vaccinated. This is a disease that doesn't pop up until you have some issues with your health, for example. And by health, I'm talking specifically about your concentrations of iron. So if you're pretty healthy and your iron levels are high, the regulator called DTXR will bind to iron, and it will repress the transcription of the tox gene. And again, that's encoded on the beta phage. But if you're in a situation where your nutrition is somewhat compromised or you're otherwise a bit unhealthy, women might be more prone to this because sometimes they have a tendency to be anemic. Then the iron levels drop in the host cell, and now DTXR is in an inactive form, and expression of the tox gene encoding this toxin will commence. The last organism that I want to talk about is Vibrio cholerae, not because Vibrio responds to concentrations of iron, but because, like Corynebacterium diphtheria, the toxin genes and other virulence genes are encoded on a phage. So these are the viruses that are specific to these pathogens, and they can move around. Many other virulence genes, as we saw earlier, are on plasmids that can also be transferred from one cell to another. So let's look at Vibrio. It exists most often as a planktonic cell. It has a single polar flagellum, and it's out in marine environments. It does associate with, as a biofilm with other Vibrio, but it can also have, as a reservoir in the environment, its association with cyanobacteria, copepods, or chironomids, the egg masses, and these are midges, which are related to mosquitoes. So in a sense, Vibrio can kind of hide out in the environments in these different types of lifestyles. Now, where it becomes a real serious problem is when water treatment facilities, even really basic water treatment facilities, are destroyed by a natural disaster such as a hurricane or an earthquake, And then there's typically a very significant increase in Vibrio cholerae in these environments, and that's when humans become infected. So when water treatment is an issue and people don't have access to fresh water, they're going to drink contaminated water and get these large doses of Vibrio cholerae. And Vibrio can then get into the small intestine and get into the crypts, and you can see that the cells are moving down into the crypts of the small intestine. Once there, what Vibrio cholerae produces is another toxin, an AB toxin. So here's our Vibrio cell up here. Here's the cholera toxin, AB toxin. The B part binds to GM1, and that allows the A, or active component, to get into the cell. And once it's in this case, we're going to see activation of one of our favorite enzymes, adenyl cyclase, and these cells that are now infected with toxin start producing a lot of cyclic AMP. And what that does is it results in significant loss of electrolytes and water from the intestine. So people become incredibly sick, not because of the Vibrio directly or per se. It's actually because of this cholera toxin and the extreme loss of water and electrolytes that results in a very watery diarrhea that these people have. So let's look at how these virulence components are regulated in cholera. So here's the cholera toxin. We're going to kind of go backwards on this. There are two separate genes encoding these different subunits. And again, that's on a prophage. Tox-T is the protein that's going to be directly involved in activating expression of this. And Tox-T, as you'll see, is a gene nearby that is regulated by Tox-S and Tox-R. I'm not going to pay attention at this point to TCP. I'm just going to focus on Tox-S and Tox-R. They're a little bit unusual, and that's why I want to point them out here. So here are the genes for Tox-R and Tox-S, so we make those components. And one of them, Tox-S, actually sits in the outer membrane. And notice that it is associated with some outer membrane proteins. So they are sensing what's happening in the environment, so that when this organism gets into the host cell, these outer membrane proteins, of course, are going to be responding to different components that are in the host. But what's unusual here is that when Tox-S is activated, because it's in this envelope layer, it will actually stimulate the dimerization of two Tox-R proteins that it associates with. And when we get the Tox-R proteins dimerizing, they can activate transcription of a number of genes, including these outer membrane proteins, and over here they're going to be involved in activating Tox-T. Now the reason that this is unusual, and this was work that was done by Virginia Miller and John Michelanis's lab at Harvard, is because normally when we've been talking about DNA binding proteins, they've been in the cytoplasm. And this turned out to be a very unusual case because Tox-R is an intermembrane protein, and yet it binds to DNA and activates transcription. So it's sort of directly sensing what's happening in the environment and what's happening in this area that we call the envelope, which is the outer membrane and the periplasmic space and the inner membrane, so that it can relay that information into the expression and activation of tox T. Tox T then activates production of the cholera toxin.